Even though my talk is not live, I'm sure you have enjoyed the lectures so far. It is also an honor for me to do the case studies in this section. Don't be surprised, I will present a roundup of previous phytolith analysis in Turkey, sampling strategies, and my previous work. It's more meant to help you understand in the first section how sampling affects the research question and what can and cannot be expected. Then in the second section, I specifically address a case study, namely the work in Zincirli. Here, I want to show how the results are or are not compatible with microbotany and the archaeological context. I hope to be able to answer your questions live at the end of the video. That will depend on the internet connection in the village of Göbekli. So keep your fingers crossed. I'm always surprised at how many papers there are on phytoliths in Turkey, and new articles keep popping up. I've tried to list here via the map from Stromberg et al., which works already exist, at least those that I know of. I would be very happy if you know of more work or if colleagues get in touch so that we can exchange ideas. As you can see, the archaeological work is concentrated on the sites around Çatalhöyük and south and southeastern Turkey. Geographical work is additionally represented in the west coast. Still, a gap seems to be in the north and the east, but as you can also see, the places where new works are planned multiply from year to year. If you are interested, get in touch. We are currently training many new young scientists. Before you start looking into your depot for samples to send us, I would like to talk about the different sampling strategies. Not every soil sample is suitable for every research question, and this is generally true for every analytical method. In particular, for microremains, I have made the experience that if you ask for samples or are asked for analysis, the possibilities are the following. You get any soil sample. This is the if there is nothing else choice. Or there is thin sections, but this is a special area that will be covered in a separate section in the workshop. Or there are different types of bulk samples from contexts and objects. But why is sampling so important? I would like to explain this in more detail. Roughly speaking, I have distinguished between the following two methods, somewhat exaggeratedly. Method 1 is take what you can get. This means that the samples are from finished excavations and there are usually less specific statements about the material possible. Method 2 is sample what you want to know. The research question can be asked before sampling. Either way, as many well-documented samples you have, as many possibilities there are. It is also important to clarify the mutual expectations between excavation projects and specialists before starting the analysis. Often we get a bag of soil sent to us with the question on the left. Our questions look something like the ones on the right. Here I show you how I would ideally take samples. This definitely includes the archaeological context, the objects, and, if possible, different samples from one context. Here I show you an example from Turkmenistan, where a stone installation was found in a room. One stone had ochre remains, but the other did not. It was unclear what these stones had been used for and whether they were still used at all in this room or were just lying there. Therefore, the room was divided into rectangles of equal size, like a chessboard, and every second one was sampled. This was not only done for phytoliths, but every floor was sampled and floated for functional analysis and macrobotany. At the site, however, an additional bag was taken for phytoliths and other samples. In this way, the distribution of the individual elements studied in this room could be traced. Then samples were taken from the stones. I took samples of these in dry brushing and wet brushing with distilled water. My idea was to see if it might be possible to detect previous use in the pores of the stones, then in the last phase of use. If the objects are smaller, samples can be taken with sonication, which can then extract the last remnants from the objects. The results showed that the larger stone was probably used in their first use as a grinding stone for grain, and later with the other stone for a non-plant activity. However, I could not determine this last non-plant activity from the plant remains, but from the traces of use. The very similar composition and quantity of phytoliths showed that both stones had stood on this floor for a longer period of time. They were probably part of the room. One idea, which I know from examining other grinding stones with ochre, is that grinding stones were often first used for grinding grain and later for ochre. However, since there were no traces of ochre on the second stone, this last phase for the large grinding stone could possibly have been a third use. Samples that have not been taken specifically for phytoliths are often samples that have been previously taken from flotation samples. This is definitely better than nothing, but I want to clarify here where the problem lies, namely in the dimension. For macrobotany, a certain volume is necessary. However, the strength of phytolith statements lies in their small scale. Results from very large samples can ultimately only tell us which plants were there, and macrobotany is definitely better suited for this. Here I would like to mention the sampling of objects, especially vessels, which were very carefully sampled, for example, in the Sirke project. Ideally, samples are taken from the surrounding area during the excavation, or in a lucky case, if it lies on a floor, as described for the grinding stones from Turkmenistan, in checkerboard sampling. But even if these have not been made, the contents are very important. It should be kept in any case, but even if the contents are no longer there, if the vessels have not been washed, you can still take a sample by brushing it off. And in the worst case, even if the vessel has been washed, you can still try to get out any remains with sonication. The probability of obtaining sufficient sample material is, of course, very high. That was a general experience report on how to deal with samples not necessarily specifically related to micro residues. But now I would like to present the specific case studies I'm currently working on. They are meant to show you what you can still get out of different sampling strategies and what you can also say with so-called negative results. We'll start with the pre-Neolithic site of Göbekli Tepe, and here I will draw on an already older presentation given at the Hilly Flanks conference in 2021 without going into details. To summarize the previous macro and micro plant remains studies in Göbekli Tepe, Reindanev, who studied macrobotany remains from approximately 2,000 liters of flotation volume, albeit with small-scale sampling by manual bucket flotation of mixed material from the fill, argues, so far there is no evidence for domestication in the unfortunately scarce plant material from Göbekli Tepe. This does not exclude the possibility that cultivation of plants had already taken place in the early Neolithic, constituting a so-called pre-domestic agriculture. Dietrich et al. Touched the difficulties of identification of domestication in phytolith research, but nevertheless, while a morphometric comparison of phytoliths from 4 samples indicate the presence of domesticated cereals and thus call Neve's results into question. Even if the sample's contexts are in some cases more specific, they rely for the morphometric analysis as well on mostly not well specified contexts and only on 4 samples. Their article has led in the scientific discussion to request if domesticated cereals may be present in Göbekli Tepe or not. Following the request that the question of domestication should be checked by further sampling, soil samples previously exported to Germany have been examined for phytolith. Although statistical analysis of this large amount of data are still in progress, I have not yet been able to detect any evidence of domesticated grains. Since I'm also analyzing samples from Chalcolithic to Iron Age samples from southeastern Turkey in parallel, I have had direct comparisons. At the moment, I have done mostly quantitative analysis in the samples. The specific taxonomic determination, which requires a separate study with corresponding morphometric analysis, will be part of the studies on the specific samples that will hopefully be done this year. However, I can summarize my preliminary results in a very simplified way as follows. Until now, there is no evidence for domestication. Floors seem to be kept clean. The fill above floors had concentration of tree material that could possibly indicate roofing. The plastering material has high concentrations of stems and leaves of grasses, possibly indicate admixture in the form of chaff. But it should be kept in mind that my focus in these analyses was not to find a hint for domestication of plants. But this morphometric issue in phytolith research, which is in my opinion a quite difficult task that needs intensive research and experience, I just want to summarize some points for the question, can phytoliths be used at all as the only proxy of domestication? I think it should only be used as a support for macrobotany, for example. And it could be possible when specialized research is done using morphometric analysis according to a standardized protocol. Since a distinction between wild and domesticated Triticum and Hordeum, wheat and barley, species require discriminant function, the morphometric parameters of different phytolith types should be included for the best taxonomic resolution. Differentiation of wild and domesticated wheat and barley was done by Ball et al. By computer-assisted measurements on scanning electron microscope images, SEM, which cannot be adapted one-to-one for light microscope images, or at least one should be aware of this when measuring microscopic particles in a high magnification. So in my opinion, it is always better to have a multi-proxy approach in the frame of macrobotany. To give you a very small insight into the difficulties of distinguishing the various wheat and barley species based on phytoliths, I would like to show you the differences between light microscope to SEM images. In the middle you can see an image from Göbekli Tepe. At the top right, a modern sample from another site. Both are light microscope images. At the bottom left and right, you can see the images from modern plant taken with SEM. What you can take from these studies for future work is, in summary, a new sampling strategy that also applies to macrobotany and includes micromorphological studies must be implemented. These are also immensely important for understanding the deposits in the settlement and the function of the buildings in Göbekli Tepe. However, to return to the domestication question, the question arises whether the extreme focus on the domestication of plants does not limit the view of research related to plant use. Are we not ignoring many equally important aspects that can contribute to the understanding of the way of life of the people at Göbekli Tepe, but also in the transition to sedentary life in the region? Do the results inevitably have to be linked to the interpretation of architecture and supraregional significance? Might it not be desirable to first understand the microhistories between humans and plants and use these small steps to approach the bigger issue of domestication? The next sites I will present and about which we will hear more talks during the workshop are part of the project I'm working on with Doğa Karakaya. It's called Plant Ways in Zero-Heatite Cities, Connections or Disconnections in Subsistence in the Northern Levant during the Iron Age. Here I would like to pick out just a few aspects about the project. For the goals, the following points can be summarized. We would like to determine artifacts and places of plant use in order to find out more about the archaeological context and its relation to social structures. Understand the developmental pathways of the political and subsistence economy in the northern Levant, and interrelationships of local climatic and environmental variations and agricultural economy. Research questions that are particularly important for my studies are: how can we merge the information to get a better picture? How to get from micro scale to the big picture? What is the place of phytoliths in this research? On the right, you see the map with the sites to be investigated. From west to east, the site of Tepe is located in the Çukurova plain, which is encircled by the Taurus and the Amanos, and Sirke is located directly east of it. To the east of the Amanos is Zincirlihöyük and Dülükbaba Tepesi, both in Gaziantep province, and further south in the Amuk plain is Tel Kainat. Today I will briefly discuss Sirkelihöyük and Dülükbaba Tepesi, and later I will present the results from Zincirlihöyük as a completed sub-project. Our general question is, which is also reflected in the examination of the plant remains, how to get from micro scale to the big picture. It is indispensable for this to adapt the selection of context to the requirements. These must be site-specific, but we need also context that can be compared between sites. That means site-specific context and context that occur in all sites. This would allow us to move from intra-site to inter-site comparison. Therefore, it is important to distinguish between different areas, structures, and features, and to cover them all as far as possible. So we need spatial, functional different areas, like citadel, lower town, temple, domestic areas, and so on. But unique structures such as temples and multiple occurring structures such as houses and households as well. And uniquely versus multiply occurring features, for example, floors, installations, pits, and artifacts. That was the theory, but the reality, of course, does not always look like this. Unfortunately, people are shaping their habitat individually. Not all sites have the same settlement structure, not all sites have exactly the same chronology, and it is not possible to cover all kinds of areas at all sites. Due to current excavation plans, preservation conditions, site characteristics, bureaucratic, logistic, and so on reasons. We have to deal with this in our investigation. Therefore, here is a very rough overview of the site-specific characteristics. It only serves to illustrate the diversity and commonalities that can be assumed, and also the prospects of how the samples will look, and it may not reflect the current state of research. For example, not all sites have the same time span, or not all sites have different areas mentioned. This, of course, reduces the context to be sampled, but also the number of samples. Sampling for phytoliths started at Sirkelehög in 2018 with the first samples of grinding stones to see how the preservation is. It continued on a small scale until 2021, when a short analysis campaign was added after the excavation campaign. This kind of fieldwork, by the way, is the most pleasant way for us who have to do analysis in the field, as we have enough time and peace to do it. However, we have to ensure that samples are taken from the context during the excavation before. For microbotany, this is usually unproblematic, as these have now become part of the everyday excavation routine. For phytoliths and other samples, this still needs appropriate advertising and persuasion, but this is also well on its way. In any case, what worked very well after the excavation campaign was the extensive sampling of objects, mainly vessels, in Sirke. In addition, it was possible to involve students in the work during this time, so that even dung spherulites analysis could be carried out. It was a happy coincidence that we also discovered starch grains, which belong to another area of microremains, which I will not go into detail today. I can say the following for Sirke. The initial situation was, for the sampling was, we had already excavated objects and we concentrated on vessel contents. The work is not yet finished and should be compared to the results of the macrobotany analysis. But I can already say that there is no evidence for cereal storage in the vessels. For the grinding stones, I can say that mostly wheat was processed. And we are waiting for the analysis of the dung spherulites, which is part of the MA thesis of Sude Gürkan, who will do the evaluation in relation to the samples from Tepe. For the future, it is important to sample floors, fire installation pits during the excavation. Sample where the context but not the objects were sampled is Dülüpova Tepesi. The extensive remains of Iron Age buildings uncovered since 2007 point to the enclosure of a sanctuary or temple. During the excavations, features were associated with deposits of burnt offering remains. Massive ash layers with many thousands of animal bones, hundreds of stamp and cylinder seals, scarabs, cibolia, and beads. Special pottery finds were dedicated to the gods of Doliche. The results of the analysis of the animal bone concluded that they were the remains of large festivals. The samples from Dülüpbaba Tepesi, after the actual archaeological work had already been completed in 2017, stem from 2019, when the former excavation area was extended due to restoration work. In this context, in continuation of the already known ash layers, an area of about 2 to 1.5 meters in size was sampled in several layers, each for archaeobotany and phytoliths in grid sampling method. The phytolith samples have revealed only a very small number of phytoliths, which indicate the use of wood. However, due to the very sparsely preserved morphotypes, no further identification of these plants could be carried out. Furthermore, dung spherulites were completely absent, whereas a high number of diatoms, microalgae, occurred. Fossil remains in the archaeological context point on the one hand to a use of wood as fuel and probably not dung for the formation of these ash layers. On the other hand, the exceedingly small preservation of phytoliths and the high number of diatoms, these microalgae, indicate that the burning process did not take place in situ and that the ash was exposed to weathering for a longer period of time, or that the fire may have been extinguished with water. These results could indicate that most probably dung was not used in a religious context, which might have been understood as hygiene perception. In addition, they offer a possible reconstruction of the events that might have led up to the final deposition of the ashes on the foundation of the temple's terminus walls. The burning of animal offerings took place at another place used for this purpose, possibly a separate installation since the ashes were homogeneous. Wood, probably high quality and good smelling, was used for the fire. The embers were either left for a while or maybe extinguished with water, until the installation had to be cleaned. In this context, the ashes were buried or disposed of along with other offerings. The objects were not burned on the walls near the temple. In addition, it can be assumed that animal sacrifices, but most probably no other plants such as cereals, were presented as offerings. In summary, I can say the following about the sampling and the results in the sites. At Göbekli Tepe, samples were used that were not specifically collected for phytolith analysis. Accordingly, the research question was subsequently adapted to the material. In this case, I decided to subdivide the analysis by context. However, the new samples taken in 2021 specifically for phytoliths from context and objects will certainly expand the previous results with more detailed information. At Sirke, the samples were systematically taken by different methods, but mainly from objects. The initial question was, what was stored in the vessels? So far, there is no evidence of grains in the vessels. For the future, we need systematic sampling during excavation of floors, and we have to evaluate the results in the light of macrobotany and dung results. At Tepe, samples were collected specifically according to the question of the use of plants in the cult in respect of offerings. The results could exclude plant, or better, grain offerings and indicate the use of wood for fuel. In general, I can give the following takeaways for sampling procedure for micro-remains. Sampling must be part of an ongoing excavation. This is not difficult, but it needs awareness and a brief training of excavators on special context, which we will do in our practical part of our workshop. Objects can be sampled later. My general suggestion for containers is keep the entire contents, never use up the entire contents for a single method. For objects in general, if possible, do not wash objects. If you're interested in conducting scientific methods on your excavation, develop questions and discuss them with specialists before excavating. Old samples may still contain information, and systematic sampling is no guarantee for sensational results, but it makes it more comprehensible. And do not forget that even there is no evidence for something special, this is a result as well. So before starting with the second part of the case studies, I would suggest to give you all some 5 minutes for a short break. So thank you.